All right, this is the second part of chapter 13 about the control of microbial growth. And we are talking about, or we are continuing talking about the physical techniques that we use for control of the microorganisms growth. So the next category is using pressure. So exposure to high pressure can kill many microorganisms. Pascalization, this is when we use high pressure, especially in food industry, to kill bacteria, yeast, mold in the food, and for maintaining the food quality, especially increasing the shelf life. We also can use the hyperbaric oxygen therapy. So imagine like the anaerobic bacteria, like Clostridia, and there are a lot of pathogenic groups in this genus. And it can cause, for example, gas gangrenes, especially in the burning patient or victims. So this bacteria is very sensitive to the oxygen, so exposure of this bacteria to oxygen can kill them. And they also can use it for wound healing and increasing like the blood vessel growth in that tissue, and stimulates the immune responses. So these are like some of the important techniques that they use at the hospitals and clinics in order to control the microorganisms and increase like the health of the tissue. And then we have desiccation. So this is the hypertonic environment that, for example, if you remember, they use a high concentration of the solute in order to cause plasmolysis, and the cells gonna lose water, and that's why, so they can get rid of those pathogens this way. And then we have lyophilization. So lyophilization, when we use like the cold with the high pressure, so like freezing in the high temperature, high pressure, so it can we call it like freeze drying, so that we use it in order to get rid of some of the microorganisms. So desiccation, so these are some of the techniques that they use it a lot in the food industry, for example, raising or making beef jerky or jam. So because it has high concentration of the salt or sugar, so it makes a very hypertonic condition, so that microorganism, they cannot survive in this condition. One reason that they add too much salt or too much sugar to our food is just for preserving it, but they don't consider like the health issues that it can cause later on. So then the next technique we have in the physical methods for the control of the microorganism is ionization. So is radiation, and we have two types of radiation: ionizing radiation and non-ionizing radiation. Ionizing radiation is much more stronger. So this is when we use gamma rays and x-rays. So they have a high energy electrons, so that they use it in order to can penetrate inside the material and can kill the microbes. So it can cause damage to the DNA, and it can also cause excessive mutations. They use it a lot in pharmaceutical and healthcare uses, and even some materials in the post office, and it can be used for the foods as well. So this is the sign that they usually use for food products, that they use ionization radiation for getting rid of or controlling the microbial growth. Then we have non-ionizing radiation that we use it, for example, in the lab. So we use UV radiation, or in the hospitals, in order to control the microbial growth. And UV is not very strong, so it cannot penetrate inside the material, but it can control the microbial growth on the surface of the item that we use in order to under the UV radiation. So usually UV can cause the cross-link that you can see it here between the thymines. So you can see it like in the DNA, these thymines, they make like a bond with together, and then it doesn't let the DNA replication and the DNA like transcription and all those RNA transcription, those processes to happen properly. And so it cannot penetrate, so we use it for under the hood for doing the sterilization of the environment that we are using for culturing the bacteria and the microorganism, or they use it in the hospital for cleaning the rooms and like the environment that they have. So if you cannot use usually heat for controlling the microbial growth or killing the microorganism, the best technique that we can use is filtration. So we can use this for filtering airs and also for filtering the liquids. For example, there are some liquids that we cannot use it under autoclave, so they are heat sensitive, for example antibiotics or blood, or there are a lot of materials that we cannot put it in the autoclave because of the high temperature that it has. So filtration, this is another important technique that we can remove the microorganisms from that liquid or from that solution easily without exposure to the heat. So there are so many different kinds of filters with different sizes that we can use for filtration. And HEPA filters, so these are the ones that they use, for example, for control of the endospores or viruses, and that they can flow in the air easily. So they filter the air using these HEPA filters. So this is a nice table for you guys from your textbook, so you can see like the different methods of the control, that physical methods of the control that we talk about. We talk about heat, cold, pressure, desiccation, radiation, and also then we talk about sonication and filtration as well. So then now you learn about the physical control. Let's talk about the chemicals that we use to control the macroorganism. So there are a lot of chemicals that we use in different for different reasons and for different objects. So the chemical we usually use to control the growth of the living tissue on the living tissue, or we can use for the inanimated objects. So they usually cannot do sterilization, but they can decrease the population, or they can remove the vegetative cells. So depends on our goal, we can use different agents. So not a single disinfectant can work in all the circumstances. So depends on what is your goal, you use different kinds of chemicals. So this is a summary of the chemical agents that we can use to control microorganism. Probably you all learn about the Joseph Lister. So he was the first person that used phenol to disinfect the treatments in the surgical for the surgical tools, and he could save the lives of many people just by sterilization of the surgical tools, because many people died actually during the surgery or after the surgery because of the infection that they got because of the non-sterilized or non-cleaned surgical techniques and tools that they used. Even phenol is very toxic to the cell membrane and the DNA, but using it during the surgery for cleaning the surgical material could be very helpful in having a much more sterilized condition for the surgery. So we're still using some phenolics such as thymol, eucalyptol, which is the natural things that they get it from the eucalyptus, lysol, and hexohex, which is they use it for surgical scrub, and also some tricolazine that probably your grandmas or grandpas they remember. So they use it antibacterial hand soap that was common before. So we can use heavy metals too, such as mercury, silver, copper, and zinc. Although some of these metals are expensive, but if they can use it for like making the equipments in the hospital, so these metals they have the antibacterial or antimicrobial characteristics that we can use it in the healthcare environment. So then we have halogens. So in this group you have iodines and chlorine, which you know it as bleach. So very common and very important agents that they use it commonly in the house or in the hospital and other like health related environments. And then we have the alcohol. So usually we use 70% ethanol. Actually 70% ethanol is much more efficient than 100% ethanol because it has water, so it can help to penetrate inside the cell and the cell membrane better. And we use it a lot, especially in hand sanitizer, and so it's a very efficient chemical agents that we use it daily. And then we have bisbiguanides that they can be bacteriostatic at low concentration but bactericidal at high concentration, and they can damage like the cell membrane, and they can also use it against the enveloped viruses as well. So, and they use it for like antiseptic for patients before surgery or needle injection. All right, so these are some of the testing techniques that we use in order to see the effectiveness of the antiseptic and disinfectant solutions or materials that we use. So one of the important technique that we use, we call it disc diffusion method. And so we're gonna spread the bacteria on a plate. So we make like a lawn of the bacteria, and then we're gonna place like a filter disc that they are soaked in different disinfectant material, and we're gonna put it in that bacterial culture, and then we're gonna incubate it usually 24 to 48 hours, and then we're gonna see like the inhibition zone that is related to the ability of that disinfectant to inhibit the growth of the bacteria. So you can see it here. So first we're going to culture, imagine the bacteria that we're going to see how this controlling or disinfecting agents, they can affect the bacterial growth. So we're gonna culture the bacteria, so and then we're gonna soak these small discs with different antimicrobial agents, and they usually have labels, so we purchase them, or you can make it, prepare it by yourself. So then you can put it on different parts of the Petri dish or the culture that we have. And then after 1 or 2 days, you can measure the zone of the inhibition, so you can see the clear area that that inhibit the growth of the bacteria there or kill the bacteria in that area. So the bigger is the zone of the inhibition, so it means that disinfecting agent is more efficient. So in this picture you can see like ATB5 is like the most efficient antimicrobial agent that we can use against this bacteria. Then the next technique is in-use test. So this is a technique that they use in order to check that that disinfectant material is contaminated or not. And also they're going to check how efficient is that disinfectant. So what they're gonna do that they're gonna add that disinfectant to a solution, and then they're gonna take 1 milliliter of that disinfectant solution, and they're gonna add it to 9 milliliter of the broth. Imagine like a bacteria broth, and then they will culture it, so on 2 different petri dishes, and then so they usually put like 10 drops on each plate. So they keep one plate at 37 degrees Celsius for 3 days, and the other one they just put it at room temperature, which usually between 20 to 22 or 23 degrees Celsius for 7 days. So if you see more than 5 colonies on either plate, so it shows you that disinfectant solution is not efficient in order to kill and to prevent the growth of that microorganisms, or that disinfectant, it can be contaminated, so they have to change it. So sometimes the disinfectant chemicals or solutions, they can be contaminated with microorganisms too. So in-use test is a very important and efficient test that they use it in order to check the disinfectant and they're contaminated or not, or their efficiency. So we are done with this chapter, and then we're going to continue with chapter 14, which is about the antimicrobial drugs. Thank you so much, and talk to you in the next chapter.